If you click this video away, then please tell me what the reason was so that I can improve. Otherwise, hi and welcome. I am trying to learn biochemistry today, carbon and its functional groups. And after we've done that, we're going to do stretching. And if you participate in the stretching, then it's validly on risk. Carbon and its functional groups. We read it partly in the previous part. Now we try to read it again and try to understand it a bit better now at the same time. Carbon has several properties that make up it particularly useful to living organisms. For one thing, it can form bonds with 1 to thousands of other carbon atoms to produce large molecules that can have many different shapes. One carbon atom can come together just with another one, or it can come together with 2, 3, 5, or 1,000. And it can have no completely different setup or shape, and can have no different functions, but it's the same carbon. This means that the body can build many different organic compounds, each of which has a unique structure and function. Moreover, the large size of most carbon-containing molecules and the fact that some do not dissolve easily in water make them useful materials for building body structures. Okay, the large size. So the most carbon-based molecules have many, so 10, 20, hundreds of carbons next to each other, and also that they don't dissolve in water. Makes it a useful basic structure for many other molecules. Now, that is what comes to now. Where other molecules can dock onto these carbon, I hope dock is an English word, can dock on. Organic compounds are held together mostly or entirely by covalent bonds. Carbon has 4 electrons in its outer valence shell. It can bond covalently with a variety of atoms, including other carbon atoms, to form rings and straight or branched chains. One carbon has 4 electrons at the outside of its shell, so at the nucleus in the middle, then 4 electrons, and it can come together with other carbons as circular, also as branched, and as chain. Other elements that most often bond with carbon in organic compounds are hydrogen with 1 bond, oxygen with 2 bonds, nitrogen with 3 bonds, sulfur with 2 bonds, and phosphorus with 5 bonds occur less often in organic compounds. Sulfur and phosphor are less often as hydrogen, oxygen, and nitrogen. So other things can bond to it. So it's a good basic structure onto which other things can stack on. I think you can think of it like puzzling something together. It's a good basic structure, and then you can build on top of it. The chain of carbon atoms in an organic molecule is called the carbon skeleton. So behind these carbons is a carbon skeleton. Most of these carbons are bonded to hydrogen atoms. Attached to the carbon skeleton are distinctive functional groups in which other elements form bonds with carbon and hydrogen atoms. Each element or functional group has a specific arrangement of atoms that confers characteristic chemical properties upon organic molecules. That is on the other side here. So different types coming together, oftentimes with hydrogen and carbon as basic structure, to form that other molecules which can do specific things in the human body. Because organic molecules are often big, there are shorthand methods for representing their structure formulas. Figure 250. In standard shorthand, carbon atoms are understood to be at locations where 2 bond lines intersect and single hydrogen atoms are not indicated. CH2OH. The H means that there are no 2 of them, but I completely get, what was it here? 2 bond lines intersect and single hydrogen atoms are not indicated. So the single hydrogen atoms are not written down in standard form to write all these chains, all these things with carbon structures or whatever, these things with structural formula. Because they're always there, and then they're also made together with numbers, but we're going to come to it later. Figure 215 shows two ways to indicate the structure of the sugar glucose, a molecule with a ring-shaped carbon skeleton that has several hydroxyl groups attached. So it's carbon ring-shaped, so there are carbon. There seems to be an oxygen. And then at the outside, hydrogen most of the times. And that is basically then glucose. Small organic molecules can combine into very large molecules called macromolecules, and macro means large. Macromolecules are usually polymers, and poly means many. Mers means part. Large molecules formed by covalent bonding of many identical or similar small building block molecules called monomers. When two monomers join together, the reaction usually involves dehydration synthesis, and that is the elimination of a hydrogen atom from one monomer and a hydroxyl group from the other. So these small organic molecules, these are a bunch of them together. These are carbon and hydrogen brought together. And these different ones of them, they can now come together to macromolecules, very big ones. So small setups coming together and building a bigger one. Large molecules formed by covalent bonding of many identical or similar small building block molecules called monomers. And when two monomers join together, the reaction usually involves dehydration synthesis. There's hydrogen by the one removed, hydroxyl by the other one. Macromolecules such as carbohydrates, lipids, proteins, and nucleic acids are assembled in the cell via dehydration synthesis. So all these things, carbohydrates, lipids, proteins, and nucleic acids, when two of these monomers come together, one gives off a hydrogen, the other one a hydroxyl. And these monomers are large formations of these large molecule formations coming together through a covalent bond. And these large formations, they are small organic molecules which are carbon and hydrogen together, set up together. I think we can still follow. I am not completely able to understand it, but I can follow. It is also possible for macromolecules to be broken down into monomers via hydrolysis. So probably the other way around. Molecules that have so one is gaining in hydrogen and the other one losing in hydroxyl, but I don't know. Molecules that have the same molecular formula but different structures are called isomers. For example, the molecular formulas for the sugars glucose and fructose are both C6H12O6. The individual atoms, however, are positioned differently along the carbon skeleton. So glucose and fructose both have C6H12 and O6. C6H12 and O6. So 6 carbon, 12 hydrogen, and 6 oxygen. That is the setup. But you are able to puzzle them a different way together. And if you build them like that together, with all the hydrogen up here, but also blue sky, you have suddenly a painting with a blue sky. And if you make it the other way around and have all the hydrogen at the bottom, you have a picture of a lovely ocean. Now, hydroxyl is ROH. Occurrence and significance. Alcohols contain an OH group, which is polar and hydrophilic due to its electronegative O atom. Molecules with many OH groups dissolve easily in water. So hydroxyl is ROH, some type of alcohol. It is negative because of the O, the oxygen. And many of them together, or molecules with many of them, dissolve easily in water. H2O, I don't really know why. And there's sulfhydryl. Phenols have an SH group. So sulfhydryl is RSH. So SH group, which is polar and hydrophilic due to its electronegative S atom. So certain amino acids, the building blocks of proteins, contain SH groups, which help stabilize the shape of proteins. So amino acids, some of them, and out of amino acids is the material out of which proteins consist. They have oftentimes SH group atoms in them, so something with sulfur and hydrogen, and it helps them to stabilize as a protein. Carbonyl, ORCR or ORCH, ketones, contain a carbonyl group within the carbon skeleton. The carbonyl group is polar and hydrophilic due to its electronegative O atom. Aldehydes have a carbonyl group at the end of the carbon skeleton. Carboxylic acids contain a carboxyl group at the end of a carbon skeleton. All amino acids have a COOH group at one end. We just said that all amino acids have carbon, oxygen, oxygen, hydrogen at the end. The negatively charged form predominates at the pH of body cells and is hydrophilic. Ester, ORCOR, ester predominates in dietary fats and oils and also occurs in our body fat, is always talking of human beings. Aspirin is an ester of salicylic acid, a pain-relieving molecule found in bark of the willow tree. And phosphate, ROO-O-, phosphates contain a phosphate group, PO4, which is very hydrophilic due to the dual negative charges, the O and the O-. An important example is adenosine triphosphate, with which transfers chemical energy between organic molecules during chemical reactions. It is very hydrophilic because of dual negatively charged oxygen. The other one was also an electronegative O. Now we're having electrocharge, now we're having the minus, so I don't know where that was explained. Now we're having an O with a minus, but the other one was also electronegative. Maybe we skipped something. We're going to come back, we're going to do it all. Amino, RHNH. Amines have an NH2 group, which can act as a base and pick up a hydrogen ion, giving the amino group a positive charge. At the pH of body fluids, most amino groups have a charge of 1+. All amino acids have an amino group at one end. Okay, whatever. We're going to read with them. We haven't understood so far that we can continue. We know what we're talking about, and I think we know enough about it, so when as soon as we don't come forward or whatever, we know to what we have to go back where we haven't fully understood or something like that. We know to what we have to go back if we don't understand. So we know basically where we don't really understand. We will be able to understand what we are lacking of understanding as soon as it comes to be a problem that we don't understand something. That was the biochemistry part. If you were here for biochemistry, you can go directly to the next part of biochemistry. If you stay here and participate in our 2 short 1 minute stretching, that is when you're on it. Having the feet sitting together, the toes to one side, the other toes, the heels and the heels. As far as you can go, toes, toes, heels, heels. And now again in toes, toes, heels, heels. But this time stay down with the head, so go into the knees. And up again. Legs split wide apart, breathe in, lift the arms up. And breathe out to one side. Again in. Out to the other one. Into the middle. Out and down. Feet together, breathe in, lift the arms up. Breathe out one arm down. Up. Out the other one down. Up. Legs split apart. Out, lean onto the knees, breathe in, out and shoulder in front, and the other one, back, out and down, bring the feet together, then breathe in, and out, come slowly with the low back up, at the top of the shoulder roll, legs apart, Breathing up, down. Out. In and up. Down. In and close the legs. And breathing out. Now it wasn't 1 minute stretching. I thank you very much and until next time.